Now, other measures, this is of course tumor volume that we're looking at, but we also often, you will have seen past press releases of our studies talk about gene expression
Yes.
analysis. And so we wanted to spend a little bit more time to really explore and explain what that means, what we're looking at.
Yeah. So this slide, this is an important. Now, I've been saying over and over, this is an innate immune activator. And what does that mean, really? Well, when we pass the targeted, when we activate the targeted hyperthermia by passing the laser light through the tumor, so one might say, well, it's just dying. So that's just cells dying, and so the tumor shrinking. So what? Because that's what does that got to do with anything? Well, what we wanted to show was that cell death. It's the name in immunotherapy is called immunogenic cell death. As the cell is dying, it's dying in such a way that the immune system takes its components and represents it to the immune system so that the immune system becomes aware of it in a different way. And so if you look at these things, we look at a panel of genes. So these are essentially the genes or biomarkers, if you will, of innate immunity. These are interferons, IFN-A1, that's interferon alpha 1, alpha 4. The interleukins that I use in the clinic, interleukin 12, 18, 1, etc. The TLR down the bottom. These are indicators of innate immune cells. So the macrophage and the neutrophils, that may not mean anything to you specifically, but what this is when you get an infection, these are the cells that will be turned on to fight the infection, and they will stay active until the infection is cleared. All the blue, if you look at the scale on the right, essentially all of the blue or shades of blue essentially relate to fail or no expression. So a dark blue is zero expression. And as you move up, if you see moving towards the green and then the yellow spectrum, these same genes are now being turned on. So what this control panel is the panel that received just laser light with no gold in the mix. And then if we treat at the tumor with our THT, the next panel will show you that we've activated so many of these cells. And so down here, the total activators, the yellows down at the TLR 7, 8, and 9, that's essentially that's a current target of immunotherapy in the clinical world. And we activate it simply by stressing. If you look at the other, there's lots of greens and yellows. So the CCL7 and CXCL10, those are receptors that are actually calling immune cells in to the tumor that weren't active on the other side. So these are all activators. Interleukin 1, and you see the yellow in the middle of it. Interleukin 1 is the number one. So it's calling in a whole bunch of other innate cells. This is just activating strong local tumor immunity that we know now translates to active immunity that can be further enabled by turning off the checkpoints, because the natural response to immunity is to check itself. And so that's where the further therapy comes from.
So just to be clear, each column represents a different mouse here, and then the various biomarkers for each. This obviously on the left being the control group, and this being the THT group. So Carmen, would it be fair to say that, let's say this second mouse treated right here with THT lit up, that presumably if we looked at the volume of that tumor, that it would have seen the most shrinkage?
Yeah, no, that's, you want to believe that it saw the most immunity. How that translated in terms of tumor volume, there's other factors to go into growth of tumors. Sometimes tumors look like they're growing because they're just getting flooded with immune cells. And so we call it pseudogrowth in the jargon in the clinics. So when we treat a tumor, the tumor looks like it's getting bigger. We say, oh my goodness, it failed. Then if you look at that tumor under the microscope, it's mostly immune cells just flooding the field because it's so strong. So we don't want to be too linear in the relationship, but what we want to say is, oh my goodness, that's Christmas. All the lights are activated, and so we're seeing good, strong immunity.
And these are stronger or more important metrics for us than just shrinking in and of itself, because this is fundamentally what's going on in the body.
Absolutely. Within this kind of data are the keys. This is where we can further tweak the immune response. So this brings us the enablers to know if we take our THT plus a PD-1 inhibitor and we link them together, can we drive it? If we take that and use an IL-2 or whatever. So this is where the biology is really sitting in that information.
Excellent.